What's up? Alexander here with Date Psychology. So today, another short video on effect sizes. We're going to look at a practical example, but thought this was worth revisiting. So effect sizes video. What are we looking at here? Effect sizes, what are they? Why are they important? So you've probably read papers and seen the way that they get reported in the media, that kind of a thing, where you will hear something is a statistically significant relationship, right? And this means there could be an association between two or more variables. There could be a difference between groups, right? Comparing one group with another. Could also mean that some kind of drug treatment, any kind of treatment, an experimental condition had a causal effect on something. So this is typically how it's going to be reported, right? Like, oh, this happened. There's an association between X and Y, or whatever the case may be. But that doesn't actually give you a lot of information. At that point, what you should be asking is, how strong is that association, right? How big is the difference between groups? Something along that. And that's what effect sizes give you. They're a measure of the strength of the relationship, right? Statistical significance, it doesn't tell you that. It doesn't tell you how strong the relationship is between two variables. It doesn't tell you how big the difference will be between two groups. It just tells you that, this is probably not something observed from chance, given your data set, basically. So what you need to look at is, what you need to ask. What are we looking at here? How big is that difference? How strong is that relationship? Because something can be statistically significant and still be, a small relationship. Meaningless, in fact. It can be so small that it, has no practical significance. And that's what we're talking about here. Practical significance is kind of what effect sizes tell you in that sense. There's a difference, there's an effect, but is it meaningful in some way? Statistical significance, the p-value, that sort of a thing, even when it's small, it doesn't actually tell you that. So keeping that in mind, let's go forward now to the next slide. So effect sizes, you can look at these in different ways. They can be a natural effect size or an unstandardized effect size. So say you do some kind of modification, you're doing ratings of attractiveness, and you say, okay, we did this to this person, we gave him a new haircut, and this raised his attractiveness ratings from 5 to a 6 on a 10-point scale. So there you have an idea. It's pretty easy to interpret. You kind of know what you're working with, this 10-point scale. And it's an unstandardized effect size, right? The unit of measurement is the original measurement used in the scale. But we often deal with standardized effect sizes that let you compare effects across groups. Sometimes an increase in a unit on a scale or something just doesn't make that much sense to begin with. You're looking at something like points in happiness. They went up 1 point in happiness. What does that mean? I don't know. It's kind of hard to interpret it. So then you'll run into measurements, like Cohen's d, Hedges g, that tell you the difference between two groups, a mean standardized difference, right? So the difference between two groups as far as the standard deviation. For example, Cohen's d of 0.5 is 1/2 of a standard deviation difference between your two groups. So that will give you kind of an example. For correlations, you're looking at the relationship between two variables, right? So you'll see here an R, right? It'll be reported as R. Pearson's R. So something like a 0.3, a weak to moderate correlation in social sciences. 0.7, a very high correlation in social sciences. You can see here in the slide an example of what that looks like. 0.3 versus 0.7, right? So, for correlations, this just goes up to 1. For measurements like Cohen's d, that can be any number, it can go above 1. But there's an example from correlations for what you're looking at. Let's go on now to the next slide. Here's an example of kind of what you're looking at with something like Cohen's d that shows you the difference between two groups, right? So in the first one here, we have a Cohen's d of 0.3. So this is pretty small difference, right? You see a great deal of overlap between the two groups. And then at the bottom here, Cohen's d of 1, right? So there's still some overlap there, but you can also see okay, most of the individuals in each group are not actually overlapping in that case. There's other measurements this can kind of tell you. We'll get into that, but let's go on now to the next slide. So a recent paper here by McKell and Shaw, they basically asked what happens when you don't share effect sizes, when you just report an effect exists, right? What do people assume when they read something like that in the media or hear it in podcast or whatever the case may be? When you say there's an effect and you don't tell people the size of the effect, they tend to assume it's a large effect. That's what it shows here. So anytime you hear this has an effect, there's this difference between groups, etc., what's really going on here? People assume it's a big difference. They assume that it's meaningful. Often, in fact, most often it's not, right? The average size of an association in psychology is like 0.2, 0.3. More often than not in psychology, when you're looking at bivariate correlations or correlations between just two variables, or even comparisons of groups for different things like high dark triad, low dark triad, you're looking at things that are actually quite small. And why is that? Because human behavior, right, it's multivariate, it's very complex. There's a lot that goes into it. A lot of the time, differences between groups or whatever, they're not actually that big. A lot of the time, one single variable just does not explain most of the variance in human behavior. So important to remember here, the important takeaway. When you don't tell people what the effect size is, they tend to assume that it's large, when in fact, at least in psychology, other social sciences, probably in most things, in fact, the effect size is not large. Let's go on now to the next slide. So we also know that effect sizes predict replication, right? You've probably heard of the replication crisis, starting about 2010, 2015 or so. A lot of old seminal papers in psychology, a lot of old effects that were thought to be real. Researchers go back, they try to replicate the original papers. They find that they can't, or that the effects are much smaller, that kind of a thing. So what do we know about effect sizes? Papers that had a large effect in the original paper, those are more likely to replicate. So when you're starting with small effects, researching something new and you're getting tiny effects, yeah, you might expect like, okay, I'm not so sure that that's going to replicate in the future, which is kind of actually what we see here. So lots of things in this slide, examples of like what predicts what's going to replicate, right? So open data. Do people share their data, for example? That's one that predicts it. So another would be the original effect size, actually the largest predictor here. Let's go on now to the next slide. So an example of this is in the research on ovulatory shifts, right? The ovulatory shift hypothesis, dual mating hypothesis, that kind of a thing. This has been a topic that's been coming up recently on social media and all of that. This is a field that we know, I should say a research line, that we know a lot of this research now doesn't replicate. So going back to 2014, there were two big meta-analyses on this. This was 10 years ago. One of them found no effects. The other said, okay, here are some effects, right? These are the results from that original meta-analysis that said, yeah, there's a relationship here, it's real. What do we see? Shifts in preferences for mates as far as masculinity goes, so facial masculinity, bodily masculinity, muscularity, vocal masculinity, behavioral dominance, all of these things, they have very small effects associated with ovulatory shifts, if they're even real, right? Remember, one meta-analysis found, okay, these are going to be real. That's what this shows. Another said no. And since then, over the next, 10 years up to this point, much newer research, high-powered studies, that kind of thing, they can't replicate the effects. But going back to the original one that said, yeah, this is probably a real effect here, what do we see? The effects are very small, right? So this is a Hedges G of 0.15 for all of them. And in short-term mating contexts, a Hedges G of 0.21. So these were never large effects to begin with. We'll talk about that a little bit in a second. Let's go on now to the next slide. I want to help you visualize here what a difference is of 0.21 Hedges G, interpreted the same as Cohen's D. So you can look here, you can see just visually that the differences in preferences for cues of masculinity, or all of these traits that were hypothesized to be good genes, preferences for those, they shift a very, very small amount during ovulation, assuming that this effect is even true. So this is a very small and a very subtle shift, okay? That was always the case. These were never large effects. What do you see here? A 91.6 overlap between these two groups. So most of the preferences that women will have when not ovulating, they're also going to have when ovulating, right? So we're looking at the control group in this case, women not ovulating, or a comparison group, in which case it would be the same women when they're not ovulating versus women when they're in that periovulatory phase, when they're fertile or able to conceive. Very small difference here. There's a 55.9% chance here that if you were to pick someone out of random from each of these groups, that the second group would have a higher score. So this is just a little bit above chance, right? That's another way to kind of interpret this and say, okay, you get these small effects. This is not something that you're going to be able to necessarily see. And that's something that Cohen said in the original kind of description of these effect sizes. Why do we call this small, medium, and large? Well, a medium effect size is typically what you should be able to begin to see with the naked eye upon close examination. So that was 0.5 as far as Cohen's original standards. In social sciences now, the average effect size for something like this is about 0.3. But whatever the case may be, this is lower than both of those, right? 0.21. So these preference shifts that occur, for women are ovulating, they have a preference for more masculine men, or they have a preference for cues of good genes. These shifts would be so small that it shouldn't be something that you can even actually observe. At least that's kind of what all of the research shows. So going on now to the next slide, short video. So this is going to be the last one. We have to ask, as I said, most of this research has not replicated. I've mentioned that many times in videos and covered all of this research in much depth in videos and articles as well. If there is a shift toward wanting traits that the manosphere has called alpha or whatever, or traits that signify good genes, as evolutionary psychologists would put it, or traits that, signal higher genetic quality, masculinity, whatever the case may be, these would be very small and subtle shifts. You have to ask yourself, is that enough to build a whole ideology around, as kind of the red pill and the manosphere has done? Probably not. These were always very, very small shifts in the past. Ask yourself, think about it. When you've heard about this topic discussed in the past in the manosphere from influencers and podcasters and all of that, or when you've just heard reports, results reported from studies in the media, did you often assume that this was a large or a meaningful effect, kind of like the paper we covered indicates people tend to do? You hear there's a difference. You just assume like this is a meaningful difference or a big difference. Very often it's not. Very often it's small. And so you also have to ask, why are red pill, manosphere influencers, that kind of thing, kind of allergic to telling you this? They will tell you, I've read this paper and there's a difference here and blah, 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 and turn it into kind of this whole ideology built around the dual mating hypothesis or ovulatory shifts or whatever the case may be. Why do they never tell you, like, this is a very small and a very subtle effect, if it's even real? Well, to admit that would probably undermine a lot of their claims, right? A lot of the idea like, oh, this is a big deal and this explains, these two types of men that we have here, the alphas and the betas, et cetera, et cetera. Given that we know effect sizes predict future replication, and these were all kind of small and inconsistent effects to begin with, it's probably also not surprising that much of this research has not replicated. Anyway, final point here, and this is what I want you to keep in mind going forward when you're looking at research and all of that. Effect sizes are kind of the meat of the paper, right? So a relationship is significant and all of that. The next thing you need to be asking is like, okay, but how big is that relationship, right? This is one of the key things that you should be looking for when you are reading the results of a paper. Anyway, hope you liked the video. Like, subscribe, hit the bell, and join the Patreon as well. We have that up. Help support the project. I will make another video for you very soon.